Hey everyone, it's photosynthesis. Let's get into this. You're gonna need some notebook paper and some pencils or colored pencils. So go ahead and pause me while you grab that stuff. And we're gonna go ahead and set the stage here. So big picture, photosynthesis converts light energy. It's not an actual reactant, it's a catalyst. Um, into chemical energy that's stored in the bonds of glucose. And it also gives off oxygen gas as waste. So not only is all of the food that we eat somehow coming from the glucose from photosynthesis, almost all of the oxygen that we breathe is coming from land plants that are doing photosynthesis as well. So what are the inputs? Well, it just requires a little bit of water and some carbon dioxide. So who does it? Obviously, plants do it. They've got these amazingly evolved structures called chloroplasts with their double membranes and lots of surface area. We also have protists that can do it, single-celled eukaryotes, and cyanobacteria, blue-green algae, which were likely the first places that photosynthesis evolved in those prokaryotes. So any chemistry teacher is freaking out right now because you have left this equation unbalanced for far too long. So what's the problem here? Matter can't be created nor destroyed. If you have 6 carbon atoms in your products, you better have started out with 6 carbon atoms as well. So we need 6 molecules of carbon dioxide for our inputs. We've got 12 hydrogen atoms in the products. So let's make sure there's 12 hydrogen atoms in the reactants with 6 water molecules. And then we've got 18 oxygen atoms here, right? There's 6 and there's 12. So we're going to need 12 here and 6 there to give us 18 as well. Okay, phew, it's balanced. So let's go ahead and set the stage, zoom on in. We're inside of a plant cell or some photosynthetic cell. And in plant cells, that's the chloroplast is the organelle that is doing the photosynthesis. So inside of the chloroplast, you remember it's got a double membrane and it's got 2 key structures that Kaylee told us about in period 1, and Natalia and Gear told us about in period 3. So you've got thylakoids, these stacked membrane discs. That's where the first part of photosynthesis happens, the light dependent reactions. The second part of photosynthesis happens outside the thylakoids in this fluid-like substance called the stroma. So if this chloroplast were once a prokaryote, which the endosymbiotic theory proposes, then this stroma would be like the cytoplasm, right? It's like the fluid inside of there. There are molecules inside of these thylakoid membranes called pigments that absorb light, special wavelengths of light in the visible spectrum. And you probably know that it's chlorophyll is the main one that we hear about. Chloro is that Greek root for green. So they appear green to us. We see them as green because they're not absorbing green. Their absorption is really low in the green spectrum, the green wavelength. So that's what they reflect. That's what we see when we look at them. Their absorption is higher in the blue range, the 450 wavelength range to the 680 to 700 wavelength range. Okay, so that's what they're absorbing and that they're reflecting green to us. All right, so let's go ahead and get started. We're going to first look at the light-dependent reactions in these thylakoid membranes, and then we'll go on to look at the stroma, okay? So this is the part you're going to diagram, is the light-dependent reactions. So do your best. It doesn't matter if you're an artist or not. You'll soon see that I'm not, but I'm doing the best I can here. So let's set the stage. This is a phospholipid bilayer, okay? So I'm not going to draw in the phosphate heads and the lipid tails, but you know that the heads face out and the tails face in. So this would be the case all the way around. And that helps us explain why we need membrane proteins to bring things in and out of this thylakoid. It's a lot like a cell in that only small and nonpolar molecules can diffuse into and out of it. Everything else is going to have to travel through a channel. So let's go ahead and draw some of these key players. First, we've got photosystems. These are the places where the chlorophyll actually is located. Okay, so there's two of them. And the first one is called photosystem 2. And that's because photosystem 1 was discovered first. So they'd already named it. Only one of the small conundrums. So here's photosystem 2 and here's photosystem 1. They both are filled with chlorophyll molecules, these pigments that can absorb light. And so we'll see how they do that in just a second. Next up are some transmembrane proteins that serve to move electrons down this membrane. So I'm just going to sketch them in here. We've got one called plastoquinone. I'm going to abbreviate it PQ. Then we've got a big membrane protein that's also a proton pump. And it's very conserved through evolutionary history. So almost all organisms have it. It's called the cytochrome complex. It's a bunch of different proteins and enzymes, and it's a proton pump. Next up, another key player in this game is a peripheral protein down here called plastocyanin. I'll abbreviate it PC. And lastly, another one, another little peripheral protein is called ferredoxin. It's got an iron cofactor, so Fe for iron. And then we'll wrap things up on the top here with a membrane protein and enzyme called NADP plus reductase. So hopefully that ASE ending is jumping out to you. It's got to be an enzyme. It's got to be catalyzing some reaction. We'll talk about what that reaction is in just a minute. Okay, on the bottom, and again, it's a little arbitrary to say where these things are. They're all over the thylakoid membranes, but in our diagram, we're going to make it nice and simple and just draw one of each. And draw this in the bottom. Is one of the most amazing enzymes and most productive ones there is. It kind of looks like this. And you know it, we've talked about it before. It's called ATP synthase. Okay, so you can guess what it does, what it's synthesizing, and we'll talk about it once we go through the process in just a second. Okay, so first, I think we've got everything on there that we need to get started. Let's just remind ourselves that out here is the stroma. That's the liquid-like cytoplasm-like stuff. And inside here is the inner thylakoid. So we're inside of the thylakoid, inside of a chloroplast, inside of a plant cell. Phew, we are zoomed in here. And these reactions are called the light-dependent reactions. All of the reactions that we talk about today are happening simultaneously. So it's kind of a misnomer to say they happen first, but we have to study them in some order. So we're going to look at these ones first. And so if you get stuck later, I'm going to ask you to walk through and explain this on your own, on your diagram. Remember, it's called the light-dependent reaction. So start with light, right? They depend on light. It's a good place to start. So the first thing in this process that we're going to see happen is sunlight hits the photosystem and it excites the electrons inside of chlorophyll. So when you did the flame test lab in chemistry, you excited electrons in some kind of salt and they jumped up into a higher energy level, but they had nowhere to go. So as they fell back down to their ground state, they gave off a color, and you saw different colors in the flame test lab. These electrons get excited, they get boosted, but they have a place to go, and that place is through what's called the electron transport chain. So I'm going to use orange to show it. So sunlight hits the chlorophyll molecules in photosystem 2 and excites the electrons. They go up to a higher energy level and they get passed off to the next protein carrier molecule in this sequence, in this electron transport chain. And so the electrons get passed along from plastoquinone to cytochrome C, cytochrome complex, and on to plastocyanin. Okay, now we're going to pause there, but just know that they're continuously getting handed off and handed off. And so what happens next is chlorophyll has a really high affinity for electrons. And so once its electron is excited and then handed off to plastoquinone, the chlorophyll wants a replacement electron and it will grab it from anywhere. So what's one of the most abundant molecules in this is water, right? So here's H2O, oxygen in the middle, more electronegative, hydrogen's on the outside. I'm going to draw two of them. And so the chlorophyll's affinity for electrons says, give me an electron. And it actually is so powerful that it can strip one from water. So the replacement electron comes from the water. And remember that hydrogen is just a proton and an electron. So if you take away the electron, you're left with a proton. And so that's H plus is another way of saying a proton. And so each of these, as the electrons get continuously handed off down the chain, each of these hydrogens in water is going to give up an electron, and the proton from it will move into the thylakoid space. Okay, what about the oxygens? Now we've talked about the hydrogen's electron, it's going into the transport chain. Its proton is separated and in the thylakoid space. The oxygens are going to combine O2 and leave as waste. Now remember, oxygen's small and nonpolar, so it doesn't need a channel. It can diffuse right out of the thylakoid, right out of the chloroplast, and right out of the cell. And that's the oxygen gas that we're breathing in. Okay, so we're now building up a concentration of hydrogen ions inside of this thylakoid space. For every 2 water molecules, you've got 4 hydrogen ions. And guess what? This proton pump is also pumping protons, another word for which is a hydrogen ion, into this space. So initially, there's a low concentration of hydrogen ions out here, but don't you worry, this is a proton pump. It can move hydrogen ions or protons from an area of low concentration to an area of high concentration. So the energy from this excited electron, as it goes into the cytochrome complex, pumps hydrogen ions or protons into this thylakoid space or inner thylakoid membrane as well. Okay, so we've built up a huge concentration of hydrogen ions. And this is really key. This is how living things do work. They make a potential, right? So by pumping these hydrogen ions in, we've now got a concentration gradient. Not only is it a chemical gradient, because you've got a bunch of chemicals, hydrogen ions, it's also an electrical gradient. You've got a bunch of positive charges here, a lot more positive charges in here than anywhere else. It's also a pH gradient, because remember, H plus is the definition of an acid. It's the H in pH. So you've built up a big gradient, an electrochemical and pH gradient inside of this thylakoid, and that is going to enable work to be done. So let's go ahead and show it while we're here, okay? This is called chemiosmosis. It's chemical osmosis. Remember, osmosis is usually water. In this case, it's going to be chemicals. So these H plus ions, protons, have built up a big gradient. They want out, they want away from each other, and their only route out, they can't go back out through the proton pump. This is a one-way street. Their only way out is, anybody? Can anybody see it? Yes, it's right here through ATP synthase. So I'll show you videos later, but this is an amazing little mechanism, rotor, that accepts the protons in, and as they fall down their concentration gradient through ATP synthase, this enzyme turns and it connects a free-floating, I'm gonna change my color here, a free-floating ADP with a free-floating phosphate. So those two go in, and you'll see this I for inorganic because it's not connected to any carbon-containing molecules. It's just a plain old phosphate. And so as these protons fall down this concentration gradient and come out, they provide the energy to turn this rotor and connect the ADP to the P to make ATP. So, woo, here we go. We've got real power energy here. So remember, adenosine diphosphate is an adenine and a ribose and 2 phosphates. And then here's one phosphate. And then we're going to connect it together. It's an adenine, a sugar, and 3 phosphates. Okay, so I just want you to see it's all there. It's just being connected together. This ATP is going to go on to the next part. The next part is called the light independent reactions because it doesn't rely on light. It relies on a different form of energy, and you just made it. It's ATP. Okay, but we're not done up here yet. We've got another product to send over to those light independent reactions. So let's show how that happens. So here's our little electron transport chain. We've passed electrons from photosystem to plastoquinone to cytochrome complex that pumped in protons, and they continued to get passed into another electron transport molecule called plastocyanin. And you know, they've gotten kind of tired. They've done a lot of work. A lot of their energy was used in the pumping of protons and in this handoff. So they're kind of almost back to their ground state now at this point. And so guess what? They need to re-energize, and photosystem 1 is their answer, because here's chlorophyll molecules again getting energized by the sunlight. And this time, the wavelength is slightly different. In photosystem 1, it's 700 nanometers. Okay, in photosystem 2, it was 680. So it's very slightly different wavelength of light. So now this electron is re-energized, and that's great because to finish off this chain, it's going to get passed into ferredoxin and then into this final electron acceptor. And in the NADP reductase, a similar thing kind of is happening as in the ATP synthase. It's going to connect nicotinamide adenine dinucleotide phosphate with the electron and with a hydrogen. So together, the enzyme NADP plus reductase, its job is to reduce, meaning reduce in charge, meaning gain electrons, and NADP plus with an electron and a hydrogen. And so we're going to end up with NADPH. Okay, so I hope you can see this molecule. I like to think of it kind of as an analogy, as an oven mitt. And it is going to carry something that's really high energy that you wouldn't want to touch with your bare hand. That is this high energy electron. So NADP is carrying a high energy electron and proton, 2 protons, to be NADPH. So the point is to carry the hydrogen over to the next part. And the carrier molecule is NADP plus. So together, the two are NADPH. Okay, so we did it. I want you to pause where you are, and I want you to see if you can walk through in 6 or 7 steps. Start with the sunlight, start with the water, and see if you can get to NADPH and ATP being made, okay? Okay, how did it go? Hopefully you're able to get through it. The second part, and this one is a lot less focused on. If you look at the standards for AP Bio, this one's kind of glazed over, it seems to me. So You don't need to know each little step. You don't need to know each little enzyme involved. You just kind of need to know big picture. NADPH and ATP are going to head on over here, and they're going to help this next part, which is called the light independent reactions. You might also hear it called the Calvin or Calvin-Benson cycle. It is also a cycle, and it starts off with a 5-carbon molecule called RuBP. So 1, 2, 3, 4, 5 carbons there. And the input here is CO2. So carbon dioxide, small nonpolar molecule, can diffuse through the stomata right up into the chloroplast. And then there's this amazing enzyme, and Gear did a great presentation on it. Check it out in the molecule, called Rubisco, that is actually going to grab the carbon dioxide and connect it to the 5-carbon RuBP. And so if you're doing your math, you've got 1 carbon here, you've got 5 here. That's going to give us a 6-carbon molecule. And you might say, great, that's glucose, we're done. But we want this to be a cycle. We don't want it to just be one and done, right? We want to keep making glucose forever. So it's going to take a couple cycles to get a precursor to glucose called G3P, and then to regenerate the 5-carbon molecule, RuBP. Again, you don't need to know all of the specifics. Big picture, you need to know that this 6-carbon molecule then gets broken down into 2 3-carbon molecules, and that takes, here's the ATP energy. Once that's phosphorylated, it's lost its phosphate group and it goes back to being ADP. So where does this ADP go? If you're saying back over here, you're exactly right. So not only is the Calvin cycle a cycle, but there's cycles between the light independent and the light dependent reactions. So send that ADP back to the light dependent reaction so it can get hooked onto a phosphate and be ATP, that then is going to head back over here and help power this Calvin cycle. Okay, we've got carbon, we've got oxygen. We're trying to make a carbohydrate, so we're gonna need some extra hydrogen, and that's where this NADPH comes in. It drops off the hydrogen here as part of these isomerase reactions, like rearranging of the molecules, adding on a hydrogen, and then what's left is back to our carrier molecule, NADP+. So where's that going? If you're thinking back to the light dependent reactions, you're exactly right. It goes back over there and grabs another hydrogen and brings it back. Okay, so keep your diagram clean if you like it. I just drew these messy arrows to kind of show how interconnected these two processes are. Once we've got 2 G3Ps, one of them will go on to make glucose, the other will go on to be recycled into RuBP. And so this cycle will continue on and on and on until we get enough food to feed the planet. So here we go. We've got the light dependent and the light independent reactions. Big picture. Walk through it. Let me know what questions you have. Again, you only need to diagram the light dependent reactions. And what I want you to do, once you've practiced it a couple times and are comfortable with it, is record yourself with your diagram walking through it step by step. So start with the sunlight, start with the water, and walk all the way through until you get to the outputs of the light dependent reactions, which are ATP and NADPH, and oxygen is waste gas, okay? So record that, and if you'll upload that to our FlipGrid, you'll see where they go. Good luck. Let me know what questions you have, and thank you guys. Photosynthesis is pretty awesome stuff, and you're in a select group if you can really understand it and explain it.